In this video, we will discuss about a paper that discusses this DANA, which is a sialic acid sialidase inhibitor, and they presented this DANA as a potential anti-diabetic treatment. So why they have presented these things is that DANA has been shown to increase glutamate in the neurons, and most of the similar observations has been also reported in the past that that this with increase in the glutamate it will indirectly cause increase in the insulin secretions. So they checked their hypothesis that whether DANA can be used as an anti-diabetic treatment or not. The first figure we'll discuss about the sialidase present in the pancreas of the mouse, in the pancreatic isolate of the mouse, and they compared two different cases. First one is the pancreatic isolate and second one is the exocrine tissues. And what they have find out is that in the case of this pancreatic isolate, the first image is the immunofluorescence image and second image is the HNE staining image. So what they have find out is that a relative presence of this new 3 mRNA on the cell surface is higher in the case of pancreatic isolate, whereas in the case of exocrine these are lower. And this new 3 mRNA corresponds to sialidase inhibitors, naturally occurring sialidase inhibitors that will inhibit on the surface of the cells, that will inhibit the sialidase activity. And the relationship that DANA has with this DANA has towards this sialidase inhibitors will be shown in this panel CDE. So in the panel C, what they have checked is that when they use DANA along with this low injection of this glucose, that is 2.8 millimolar, they do not see any increase in the insulin, whereas they see the decrease in the insulins, which is not significant. Still they see a decreasing trend with the DANA concentration of 0, 10 and 100 micromolar. However, when they increase the amount of glucose and then they use the DANA in far lower concentration, they see that there is an increase in the amount of insulin formations, that is for 0, 1 and 10 micromolar, they see that increase in the insulin formations. So from this they made a point that DANA itself is not sufficient to increase in the insulin levels. However, when it is combined with the increased glucose levels, then it can help in increasing the insulin levels. This is what the conditions occurs in the patients who are affected with the diabetic, that is they have high sugar level. Whenever they have high sugar level, they should their body should secrete more insulin. So this DANA can help the body to secrete more insulin in the conditions where high glucose are found. And after establish their relationship with the insulin in the presence of glucose, they further checked the sialidase activity, the DANA sialidase activity in that conditions, and what they have find out is that with around 100 macromolar, the sialidase activities of the DANA is statistically significant, and even with 10 macromolar they see the statistical significant DANA activity. And when they check this DANA activity in the mice models, they see the similar trends, that is the amount of nu3 is significantly goes down whenever this DANA mouse is treated with this DANA. So indicating that this in the mouse case we do not see any sialidase activity. So this indicates that this DANA seems to be working properly even in the mouse models, and same things are reflected in the in vitro models. Same things are reflected in the in vitro models. So all this indicates that this DANA with its natural functions, which is functions towards inhibiting sialidase activity, it will also help in enhancing the insulin levels when complemented with this high glucose concentrations. So in this figure they have made the 2 points, that is DANA's has two functions. First, it will inhibit the sialidase activity that is already established in the previous literatures. At the same time, it will also helps in increasing the insulin concentrations in a conditions where high glucose are present. Whereas when no high glucose are not present, they do not seem to affect too much on the insulins. That is the reason that they uh, presented the dana as anti-diabetic treatment. So this is just a first figure. We will discuss the other results in the subsequent videos. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.